Hello and welcome to our event here for the Cambridge Festival. We're broadcasting live from the Gurdon Institute in Cambridge today. And we're going to tell you about how tiny mutant worms help us study human development and disease. So I'm Claire O'Brien, Information and Communications Officer at the Institute, and I'm your host for the event. I just want to remind you again, we are recording the whole event. But as far as we can tell, your names and the questions and so on are not going to be visible during the event or in the recording. So let me tell you about what we've got lined up for you. First of all, we're going to start with a video tour of the Institute that will last about 8 minutes. Then we'll move to the main presentation by our fabulous PhD student, Anna Townley. That will take about 20 minutes. So then we'll be around 2:30. We'll open up to your questions to a panel, because we're very, very lucky today. We've got 2 more scientists joining us for the panel, and they are our director, Professor Julie Ahringer, and our deputy director of the Institute, Professor Miska. So you'll be able to put in questions using the Q&A widget at the bottom of your screen, both about Anna's presentation and then more broadly about the work of the Institute and in the labs of Julie and Eric. So without further ado.
Welcome aboard our Institute tour. I'm Bruce Daniels, and I'm going to give you a flavour of our exciting and important research, showing you where and how it all takes place. Our purpose-built building in the heart of Cambridge houses 16 research groups, and along with core support staff, we have about 250 people on site. The researchers here publish around 80 papers per year in top scientific journals. It was John Gurdon who established the Institute almost 30 years ago to bring together researchers working on both developmental biology, how a single fertilized cell gives rise to a whole organism, and cancer biology, what happens when normal processes break down. Dr. Gurdon won the Nobel Prize in 2012 for proving that every cell in the body contains the same genetic material. John still works here in the lab every day. Our research groups all follow different but related fields so that we can describe our work in 4 topic areas. Let's head upstairs to the St. Johnston lab, where researchers use fruit flies, Drosophila, as a model organism that shares 70% of the genes that cause disease in humans. One of the techniques we specialise in at the Gurdon is microscopy, to see things happening on a minute scale, such as these cell linings of the stomach of the fruit fly called epithelial cells. Here is one of our confocal microscopes in which we illuminate a slice of the sample. The fluorescent colours can show cell nuclei, genes, or the structural scaffold inside the cell. We can track what goes wrong if we manipulate the genes in the flies to give us clues about what might be happening in human cancers and other diseases. Some of our researchers are actually building the next generation of microscopes for super resolution microscopy. Fruit flies are used in several labs here for investigating different aspects of health and disease. One of these is the study of mitochondria, the power plant of cells. The Mar lab uses genetic manipulation and breeding experiments to track how mitochondrial genomes compete, providing insights into human mitochondrial diseases. The Brand lab is another that uses fruit flies. This time to examine the stimulus for brain development. A different model organism is the nematode worm, just 1 millimetre long, and easily grown in large numbers in Petri dishes. The Ahringer lab researches how genes are turned on and off in the three-dimensional structure of chromosomes called chromatin, packed into cell nuclei. The Miska lab focuses on how RNA, the working copy of DNA, can affect gene expression and inherited characteristics. Frog eggs provide a model of a cell that is large and plentiful, and is still used by the Gurdon lab, and also by the Gallup lab, who are exploring cell membranes and their finger-like structures called filopodia. Yet further model organisms, including yeast, are researched in the Zegerman lab. Whilst human cells in culture are researched in the Jackson lab. The Serrani lab works on the earliest stages of human development and the germline, requiring special culturing techniques. A new specialism at the Institute is the culture of organoids. Tiny clumps of self-renewing cells from specific organs such as the lung that provide a way to observe development in progress. The Rawlings lab are investigating lung development and regeneration in the hope of finding clues to help premature babies and adults with chronic lung disease. Some types of research generate huge quantities of data that require storage and then semi-automated analysis, such as the results from DNA and RNA sequencing, which our bioinformatic experts will work on. The specialist imaging processes also turn out tens of terabytes of data. For all of this, we need dedicated high-performance computing clusters to store and process the data. At the center of the Institute is the all-important media kitchen, where the team prepare all those Petri dishes, fly vials and food, and dozens of different types of chemical solutions used in lab experiments. Having this dedicated service means the scientists can concentrate on their research and can rely on consistent, reproducible reagents and growth media, a luxury for most departments. When researchers are not busy in the lab, our tea room buzzes with people in discussion over coffee and cakes or having team lunches, often spilling out into the garden in good weather. This also provides a space for large seminars and parties. The admin team support the scientists with day-to-day paperwork, submitting grant applications, and sorting out visas for our researchers who join us from all over the world. Back in the basement, our building and services team keep everything running. From gases to waters, to chillers to heaters, to washers and to autoclaves. Finally, the storeroom has all the supplies any lab could desire. I'd like to thank you for joining us on this tour. A tour that supports our public engagement program, an important element of our work. Our mission is to make our fundamental biological research accessible and responsive to the public for the mutual benefits of inspiration, knowledge exchange, and trust. You've now had the opportunity to see from the inside all our research. And I hope you'll keep in touch, following us on social media and looking out for us at other public engagement events. Thank you again for taking this tour.
Sorry. I hope that's given you an idea of the hive of activity that would normally be going on in the building when all of those researchers are packed into the labs and busy. So let's get straight on to Anna's presentation. And again, please do type in any questions as they occur to you for Anna, and we'll review those straight afterwards. So I'll share my screen again.
Hello, my name is Anna, and I'm a PhD student at the Gurdon Institute, where I study in the laboratory of Professor Ahringer. And today I'm going to talk to you about how mutant worms can teach us about our own embryonic development. So at the Gurdon Institute, one of our strengths is that we work with a variety of different model organisms in order to understand human development, and each one of these organisms has their own strengths and weaknesses for different biological experimental techniques. In our group, we work with the model organism Cenorhabditis elegans, or C. elegans. C. elegans is a nematode worm which can normally be found in rotting fruit, where it feeds on bacteria. The fully formed adult worm is approximately 1 millimeter in head to tail, and you can see an image of it on my slide. Today, I'm going to tell you a bit about how C. elegans became a model organism in biology and led to some Nobel Prize winning discoveries. And I'm also going to give you some information about how we use C. elegans to study development in our group. So the nematode worm was pioneered as a model organism in 1965 by Sidney Brenner, who was working at the Laboratory of Molecular Biology in Cambridge at the time. And this went on to him winning the Nobel Prize in Physiology or Medicine in 2002, along with Robert Horvitz and John Saulston. And this Nobel Prize was won because they developed an understanding of development of animals through work in C. elegans. So what do we mean by development? Well, in biology, development describes a process by which a single fertilized egg or embryo undergoes changes through cell division and movement of cells to produce the fully developed adult organism, which will have all of its organs and tissues in the right places and carrying out the right functions. This process is highly complex and is very regulated by biology through the expression of genes in the right places at the right times. It's a very complex process that has been interesting to biologists for centuries. And by studying it in simple model organisms such as C. elegans, we will hopefully be able to understand it in complex organisms such as ourselves. So you can see here an image from the lab book of Sir John Saulston when he was working on the Nobel Prize winning work back in the 1980s. John Saulston would spend 8 hours a day sat at a microscope in a dark room looking at C. elegans under just a simple light microscope. Each day he would draw diagrams of what he saw, and he would pick a specific cell and follow where it moved within the embryo and which cells it divided to produce. And he would draw these hand-drawn diagrams with each circle representing a different cell. And he would use 3 different colours, black, red and green, to represent the different layers in 3D space of the embryo. After 1.5 years of research, this led to these diagrams, which show the positions of cells within the embryo as it develops to the larva, and which cells will produce which eventual cells. This work led to the pioneering production of the cell lineage map for C. elegans, and this can be shown here. The cell lineage map shows from the very beginning of the organism which cells go on to produce which cells in the adult. For example, here you can see in green highlighted all the cells which go on to produce the pharyngeal muscle of the worm, which is found in the head. So John Saulston and others' work led to the full, complete mapping of the lineage diagram for C. elegans. And this can be done because C. elegans is a particularly unusual animal in that it is what is known as utilic. Utilic means that its lineage diagram is the same every single time. All adult hermaphrodite worms have exactly the same number of cells, and the pathway of development is exactly the same every time. It's amazed scientists how this precise process can happen so reproducibly every single time. So this work led to the first complete animal cell lineage in 1983, and the first complete wiring diagram of a nervous system in 1986. Furthermore, in 1998, C. elegans was the first animal to have its genome sequenced, and this was also done by Sir John Saulston, who later went on to work on the Human Genome Project. You can see here an interview by Boris Johnson, our Prime Minister, when he was a journalist at the Daily Telegraph in 1998, and he was interviewing Sir John Saulston about his work on worms. And in this article, Sir John Saulston describes C. elegans as a microcosm of humanity because it has a gut, a nervous system, muscles, and reproductive impulses just like human beings. And what's more, it shares 40% of its genes with us. This is striking because although you might think that C. elegans is a very simple 1 millimeter long nematode worm, much different to a human being, we actually share a large amount of our genetic material. So why is C. elegans such a useful model organism for studying biology in the laboratory? Well, as you can imagine, it's very much more simple than a human being. Whilst humans have hundreds of trillions of cells, a C. elegans worm will have about 1,000 cells in each adult. It also develops really quickly. So an adult worm can develop from embryo to adult in approximately 3 days. And over the course of 2 days, each hermaphrodite adult can lay over 300 eggs. This means that in the lab, we can generate millions of worms in just a few weeks in order to study them in the lab. In addition, C. elegans is often found as hermaphrodites, and this means that a single worm produces both the eggs and the sperm, so they're able to self-fertilize. This means it's really easy to make genetic mutants. C. elegans are also transparent, and this is great for microscopists because we can look under a light microscope and see the internal organs of the worm without having to carry out any manipulation or inject dyes into the worm. Finally, it's really well characterized because it was developed as a model organism early on and led to some really important discoveries. Scientists such as myself who are joining the research on C. elegans have a wealth of information on its development and genome that we can build upon. So by using this microcosm of humanity, we can hopefully understand the fundamental processes of development of human beings in a much more simple model organism and then work up to understanding more complex organisms. You can see here on the right hand side an image of a plate that I grow my worms on in the lab. So these are grown on agar plates, and on the surface of the agar plate we seed a lawn of bacteria or E. coli, and this layer of E. coli serves as the food and the C. elegans worms will swim through it. They develop very rapidly, and you can see here the adult worms, the very thick and dense worms. The little tiny circles on the plate are embryos laid by the worms, and you can also see some small larvae which have hatched. Another Nobel Prize winning work, which was carried out in C. elegans partly, was the introduction of fluorescent proteins as biological markers. And I wanted to introduce this to you because it's a really interesting and useful tool in biology to work out the functions of genes. So this work began when scientists in Japan identified and extracted the fluorescent molecule from jellyfish. And this is known as green fluorescent protein, or GFP. Since then, scientists have modified this protein to produce a variety of different fluorescent coloured molecules, and they've introduced them to lots of different biological animals that we use in the lab. So you can see here a C. elegans worm, Drosophila, or the fruit fly, rabbits, zebrafish, rats, and different human and plant cell systems that we use to study biology. The introduction of GFP as a fluorescent molecule to label cells is really exciting because we can use it to find out the function of genes. We can make sure that this fluorescent molecule is only produced by cells that express a certain gene. And by looking at where these genes are expressed, we can deduce their function. For example, you can see here the cover of Science, which is a scientific journal. And in 1994, C. elegans was the first model organism to have the green fluorescent protein introduced. You can see here they've managed to light up only the cells, which are neuronal cells, with GFP. And so these cells can be seen under the fluorescent microscope. Here is another example. In this microscope video, you can see worms which have been genetically engineered to produce GFP, the fluorescent molecule, only in cells of the hypodermis, which is the outer skin layer. Each individual spot on this video represents a single cell producing this fluorescent molecule. There are also worms on this plate which are producing the fluorescent molecule in the pharyngeal muscle in the head. So what do we do in our lab? Well, in our group, we're interested in how genes are regulated during the process of embryonic development and larval development in the worm. One of the things that made me really excited when I first joined the lab was watching a simple worm under the microscope and being able to see the development from egg to embryo. So here you can see a worm swimming across the plate, and you should be able to see the internal organs. So here is the gonad, and here is the intestine. If I pause it here, you can see what is known as the gonad, which is the organ that contains the sexual reproductive cells of C. elegans. And this runs along the length of the body. You can see here these large rectangular cells, which have large circles in them. And these are the egg cells, the unfertilized eggs, and their nuclei. As the cells move along the gonad, along the length of the worm body, they grow larger, and eventually they reach the spermatheca, which is where the sperm are stored. Here, the egg cells are fertilized to produce embryos, which divide to produce more and more cells before they are laid here through the vulva onto the plate, and eventually they will hatch. As a young scientist, it was really exciting to be able to see in a live organism as it moves across the plate this whole process of development from an unfertilized egg to an embryo that will eventually develop into a full adult worm. So what am I studying in my PhD project? Well, I'm really interested in understanding how cells regulate their genes, and I'm looking at that during development of C. elegans. You can see on the right here an image taken down the electron microscope of a nucleus in a cell. Electron microscopes can achieve much higher resolution than light microscopes, so we can look at such tiny, tiny structures inside cells. This picture was produced back in the 1970s, when they discovered that the DNA inside the nucleus forms two different types. You can see here the white DNA, which is very loose and accessible. And this can be accessed by the cell and uncoded. So the DNA is uncoded and leads to the production of proteins which will carry out the function of the DNA. In comparison, there are these darker regions of DNA. This is known as heterochromatin. These dark regions of DNA are highly condensed and really inaccessible by the cell. And this means that they are not encoded and they do not lead to production of proteins. So effectively, they are turned off by the cell or silenced. Why would a cell need to turn off some genes and turn on others? Well, there's two reasons for this in development. Firstly, each cell has exactly the same genome in an organism with the same genes, but they all carry out different functions, so they need to express a different set of genes. For example, a stomach cell might need to express the enzymes needed to produce stomach acid, but it doesn't need to produce the protein receptors for neurotransmitters that you might find in the brain. So by controlling its gene expression, a cell can have a specific function suitable to where it's found in the body. Furthermore, during the evolution of animal cells, viruses have inserted their DNA into our cells and it's integrated into our genome. And this has led to a battle between the animal cells and the viral DNA. Animal cells have evolved the ability to turn off viral DNA to prevent it from causing any more damage. And this means that we've developed this ability to silence DNA. So both of these processes, regulation of genes in order to carry out a specific function and in order to turn off viral DNA, are important for the normal development of a healthy organism. So In order to understand gene silencing, I've been using another technique which was found via Nobel Prize winning work in C. elegans, and this is called RNA interference, and it's a tool that we use to turn off genes in the lab. It's also a natural phenomenon that occurs in biology. It was first discovered by scientists Andrew Fire and Craig Mello, who found that when they injected a molecule known as RNA into C. elegans, the worm behaved as though it was lacking the gene which had the same sequence as that RNA molecule. It turned out that they could also get this to work by feeding worms with bacteria that contained the RNA molecule. And when the bacteria were broken down by the worm, the RNA was absorbed and led to the same effect. As a result, scientists have been able to manipulate this natural process by feeding worms specific RNA molecules, which lead to turning off of certain genes. And this allows us to look at their function. Because if you look at the effect of a gene, the effect of a lack of a gene, you can understand its function in the way that by looking at what happens in human disease, we can understand the normal processes in healthy individuals. In our group, before I arrived, they developed a whole library of over 19,000 bacterial strains, each of which produce a different RNA molecule, which targets a different gene in the cell and turns it off. So we now have this library that's able to knock down or turn off every single gene in the worm genome, one by one. How am I using this amazing tool? Well, I'm using RNAi to turn off genes and look at the effect on their growth and development of worms. I'm doing this in two different types of worms. Firstly, the wild type or healthy worms, and also in mutant worms, which show defects in their turning off or silencing of their genome. I look at the effect on the growth, fertility, and morphology of my worms. And if I see a much stronger effect in the mutant worms, unable to do silencing, this suggests that these genes that we are knocking down or turning off by RNAi are also important in this process. So day to day, this involves a lot of moving worms around and looking at them down the microscope. So I start off by generating a large number of worms and creating plates for them to grow on. So you can see here my 6-well plates, which contain agar and a lawn of E. coli in each well on which the worms will feed. On each of these wells is a different strain of E. coli, which produces a different RNA molecule, which is able to knock down a different gene. By making all these plates, I can screen thousands of genes at a time. We then transfer the worms to the plates by using what is known as a biosorter or a worm sorter. And this is much quicker than doing it by hand. This worm sorter is able to count exactly 3 worms per well of my plate and also measure their size. And this allows us to control the age of the worms used in our experiments. The way by which it works is you place the worms here in the sample pot in liquid, and the worms will pass in the liquid through the tubes in the biosorter. And in these tubes there are lasers, which will measure the density and the length of the worms and also count them. And it uses airflow through the pipes to divert worms either onto the plate or down into the waste tray so that it can count and measure worms and dispense them for me. After this, I will incubate the worms on the plates until they have grown to adults and produce offspring. And then we look at the phenotype of the offspring. The phenotype is just the observable characteristics that I can see down the microscope, as well as looking at whether it's a slow or fast-growing mutant or whether it shows defects in fertility. We also see some changes to the morphology or the shape of the worms. So, for example, a lot of genes that cause defects in gene regulation will also affect growth of the worm. So you can see here 2 wild type or normal worms. And here is a phenotype we call dumpy, which is when the worms are short and squat and they're kind of unable to move properly along the plate. Opposite to this, you can also get particularly long worms. Other gene defects cause effects in ability of the worms to move, so they can become curled up on the plate or paralysed and unable to move. Finally, another effect that we see is defects in vulva development. Development of the vulva is really important during adult development and is very well controlled by signaling and gene regulation. You can see here some worms that have a protruding vulva on the side of their body. Another particularly grim end for the worm is the ruptured phenotype, which happens when the vulva doesn't develop properly and the intestine actually explodes out of the vulva. And you can see here the intestine outside of the worm on the plate. So by looking at these phenotypes, I will find genes that when turned off by RNAi have a stronger effect in my mutant than the normal wild type worms. I then hope to go on to characterize what the role of these genes is in development. So I hope I've convinced you today that not only is C. elegans a really exciting model organism to work with in the lab, it's also very powerful for us to understand human development. Hopefully, work in C. elegans will later go on to us understanding development in humans and development of drugs and therapeutic approaches to help treat disease in human beings. So I'd like to thank my lab, the Ahringer lab, and my supervisor, Julie Ahringer, and also Rupali Pradhan, who I work on in this project. I'd also like to thank the Gurdon Institute media facility, who helped me out with all the huge number of plates that we use, and also our funders, which include the Wellcome Trust, the Medical Research Council, and the BBSRC. And I'd also like to thank you for listening.
Thank you, Anna, for an absolutely fascinating talk. So we can now bring in all of our scientists for the panel. Anna, do put your camera on, that's fantastic. And Julie and Eric. Julie, welcome to, let me stop spotlighting me and let's hope we can get everyone else on to the view. There we go, that looks better. Julie, please, welcome to the event. Would you like to say a bit about your research?
Yeah, so hello, welcome everyone. It's a pleasure to be here. Anna's given a very nice introduction to what we're studying and what a great model organism C. elegans is. And just to emphasize that we can really make advances studying C. elegans because the genes that are in this small worm that takes only 3 days to grow up are very similar to genes that exist in the human. And the work that we do in my lab are really to understand how are the instructions of the DNA sequence read to transform a one-cell embryo into an animal. And that happens, all animals do that. And it comes down to how we turn the right genes on and off, as Anna nicely articulated. We do this for two reasons. One is it's really fascinating to understand how this all happens. It's miraculous. But also it's relevant to human disease. A lot of diseases we know are caused by gene expression being abnormal. Either the right genes don't get turned on or the wrong genes don't stay off. And so understanding how genes turning on and off help us understand disease in that way. But also we now know that some diseases are also caused when programs that are turned on in the embryo are abnormally turned on in the adult. And that's also important for these, you may have heard about reprogramming. So understanding how you keep the embryonic types programs off later in development is also important, and we can study that in the worm.
Thanks, Julie. And Eric Miska, welcome to the event.
Hello. Yeah, I also would like to say awesome to be here, and it was great to hear Anna's talk, and I think she really explained what this little worm is all about. We are studying C. elegans with a particular interest of what the genes make. I mean, genes in worms and genes in humans. And genes make a molecule called RNA, and then the RNA goes on to make the stuff that our cells and worm cells need, proteins mostly. But it turned out that in C. elegans, it was first discovered by Victor Ambrose and colleagues that some of this RNA stuff is itself active, and it helps cells decide what to do. Important decisions during development and also relevant in human disease. So RNA, more than just an intermediary between the gene and the sort of final product, maybe the protein, RNA is used for decision making in the cell. And as I already mentioned, some key discoveries were made here in C. elegans. So we're studying regulation of cell divisions of the level of RNA. And it turned out that some of these genes that make these regulatory RNA are very important in human disease, in particular cancer, neurodegenerative diseases, and also metabolic diseases. And as Anna mentioned already in her talk, there is a very useful technique that emerged from C. elegans where you can switch genes on and off using a technique called RNAi. So you can use artificial small RNAs to switch genes on and off, which was the Nobel Prize winning discovery for Andy Fire and Craig Mello. The question might be, you know, how does this work? Where does this come from? And again, this is related to the sort of regulatory power of RNA, and it's used also in different ways. It turns out then when C. elegans and a lot of animals and plants are infected with viruses, they use this kind of technique to silence not themselves, but to silence the viruses that infect them. So RNA is a powerful tool not only in gene regulation, but also in the fight against viruses. If that's true in humans, it's less clear, I have to admit though. Finally then, this RNA stuff is quite powerful because it can also contribute to heritable information. So we are very familiar that our genes are made of DNA, and the DNA stuff is probably passed on through generations. Turns out RNA can also be passed on from generation to generation, and that contains information. And in C. elegans and many other animals and plants, this is very important, and very interested to understand if this is also relevant in humans. So that's it more or less in a nutshell. We're studying RNA, taking advantage of all the powerful tools that Anna described in C. elegans. Thank you.
Thank you, Eric. So I'm going to start looking at our list of questions. Got lots of questions coming in. The first one, here's quite a tricky one. I don't know who's going to launch in and take this one. Are utilic organisms, which Anna mentioned the nematode worm is, relevant for non-utilic organisms? Who wants to take that one?
I can give it a go. I was going to say, so utilic organisms are very rare, and they're mostly very like microscopic organisms, and C. elegans has like closely related organisms that aren't utilic. But I think the degree of similarity in terms of how they function, how their cells divide, how they develop between organisms that are non-utilic and utilic is very similar, and it doesn't stop you being able to gain insight from that. I don't know if Julie wants to add to that.
No, that's a good answer.
Okay, the next one then. Here's a really broad one. What will C. elegans contribute in the next 50 years?
Here's one for Eric.
My God. Well, I guess what is so amazing.
A lot.
Sorry, I'm not evoking God
can
to
you
help me
start?
out with the answer, but instead, I would say that, you know, C. elegans has been around as a model for, I don't know, 50 years or something like this, and given us a lot. I think a main focus has been to really understand how genes give us, you know, essentially a whole organism. And I think going forward, I think what will be very exciting to figure out how genes and interaction with the environment really shape an organism. And there, again, this really simple and straightforward model in lab, C. elegans, has a lot to contribute. Yeah.
I can add something too. in the work that we do, and also what Eric works on. Really understanding how the genome is functioning at a very precise level, the way that C. elegans develops very rapidly, and because we know every cell type as you go through, really will allow us to understand these very precise processes that go from one cell to the next, to understand how the daughter cells differ from the mother cell, and to look at the precise pattern of reading the instructions as you go through cell to cell.
Okay, how about this one? Again, it's relatively broad. We share 40% of our genes with C. elegans, you said, Anna. How does this compare with, say, fishes and primates?
I should know this because this is one of the questions I did on our family Zoom quiz over this year. Because I think people are often surprised by how much DNA we share. So I don't know about fishes, but I think primates, like the closest ones, we share 99% of our DNA. And that's really surprising how different they can be, yet how much we share. But I'm not sure about fishes. How much DNA does cichlids share, Eric, with humans? Do you know?
I don't know on the top of my head. What I would say, though, is that you have to be careful what exactly you're asking, because, you know, our genomes are big. And as Julie said, we still don't even understand all the stuff that's in there. And the genes that are sort of sitting in the genome, they make up quite a small fraction of the genome, in particular in organisms like us or fishes. So, and as far as we know today, most of the genome might not have any current function at all, but might be a historic artifact and will tell us where a genome might be going in the future in evolution. So I think sharing how many genes are shared might be more relevant. And I think, Anna, you described very well that even with the worm and human, we're sharing 40% of our genes, which seems incredible.
And also, if you take those genes, almost invariably when this has been tested, if you take a human gene and you put it in and replace the worm genome with the human gene, that gene can function appropriately in the worm. So not only do these genes look the same, but we can see that they do have very similar functions.
Okay, how about this one then? Another broad one. What are the ethical issues you have to face while doing this research?
Mm-hmm.
I guess there are not as many working in C. elegans because it's a very simple model organism and we don't have to worry about things like the Human Tissue Act, but we do create mutants, genetic mutants, so we have to make sure they're destroyed and don't reach the environment where they could spread.
That's
right. Yeah, maybe if I could just add a little anecdote, since you mentioned John Saulston, who shared the Nobel Prize for figuring out exactly how all this cell division and differentiation is happening in the worm. The story goes that, you know, once he studied an individual from an egg to a whole adult animal, that afterwards he wouldn't just kill the animal or throw it in the bin, but he would put it out to pasture, put it on a fresh plate petri dish with lots of bacteria, so it could live its life happily after having contributed so much to understanding of developmental biology.
Because, I mean, essentially these worms are out in the environment, they're in the soil in your garden and so on anyway. So yeah, there's nothing too frightening about them from that point of view.
Yeah, probably you can order them from your garden center. I'm not a gardener myself, so I don't have any detailed knowledge, but you can order them as a whole pack to add them to your soil or to your compost heap. Yes, and you will casually kill many of them by just using general gardening, I think.
Mm-hmm. So how about actual discoveries that have been made about human biology using the worms? Have we got some examples we can share from our research here?
Well, certainly, if you have the human, it's really a combination. If you take the vulva development, that's controlled by a signal, what's called a signal transduction pathway. That's called the RAS, this central protein called RAS. And this whole pathway, the genetics of this pathway was worked out through work in C. elegans looking at vulval development. And in parallel, very interestingly, in Drosophila, looking at studies of the fly eye, also is controlled by the same signal transduction pathway. And this pathway controls many processes in humans, and it's also, when it goes wrong, it causes cancer. It's one of the major cancer diseases, cancer genes is the RAS gene. So understanding this RAS signal transduction pathway with seminal work from C. elegans and Drosophila has been really important. That's in understanding this cancer by RAS.
Mm-hmm. Eric, any examples from your lab?
Well, maybe another classic example is the discovery of programmed cell death. So during development, a number of cells in our bodies are programmed to die. For example, if you ever look at the feet of duck, you will see that they've got this webbing between the fingers. Most of us won't have webbing between our fingers, but during development, there were originally cells there, but they were programmed to die. And how this works, this is not an accident, this is part of normal development, and how this works, the genetics thereof has been worked out in C. elegans first, and it's very important in cancer, neurodegenerative diseases, and trying to understand the immune system fully, for example.
Okay, can RNAi turn off genes in humans too?
Yes, it definitely can. I think it's definitely been involved in some clinical trials for treating human disease. The only problem is trying to get this RNA molecule to the human cells, because when we're feeding worms, we feed them bacteria that make this RNA, and it can just diffuse from their intestine into all of the different worm cells. But obviously, human body is much bigger, and they can't just diffuse straight into the different cells. So it's harder to kind of make such a big change in expression of the whole human being.
Fair enough. So here's a very pertinent one for the moment. How has our lab-based research been able to continue over the past year, and what changes have we made in the lab in order to be COVID secure? Julie.
There've been many changes we've had to do. And really, in the beginning, we actually had to lock our building down for 3 months. And the only things that we kept going were to keep our animals alive and kept them fed and safe, and our reagents. And we had a small team, very dedicated, that helped us do that. And then we had to, in reopening, we had to change all of our procedures, have 2 meter social distancing. You can see none of us are wearing masks because we're all in single person offices. So, but everywhere else, if I leave my office, I have to wear my mask, and it's here waiting for me. So when I leave, we have we've rated every room with the number of people who can be in it, and we have to work on shifts rather than working full time. So unfortunately, we've had to slow down our research because we can't fit in everyone with 2 meter distancing. So people are coming in early, working late. We've managed, and everyone is really dedicated to try and move the research along. That's been really I'm really proud of the Institute and all the members, because we've really been still achieving a lot, but unfortunately it has slowed things down. But we're doing well.
Okay, here's a probing one here. So given the advances in molecular techniques, do we need to study model organisms today? You know, could we not go straight to humans in some way?
Well, that's,
yeah.
Can start, and then
I was gonna say that, like, you need a kind of, obviously, like from my presentation, I was saying that humans are really complicated to study. Also, there's obviously huge ethical problems. We can't go around genetically mutating humans. But also, by looking at the kind of more simple organisms, we can grow them much quicker. We have techniques that we've already established to look at them. And because of the degree of similarity, it's really useful to do these experiments more quickly and easily, and then go up to studying more complicated organisms. I was just reading the question, and it's asking about kind of using new model organisms and things. And I think we choose model organisms because they are particularly useful in terms of the tools that we can use them for, and also because we've got this broad expanse of past knowledge that we can build on. But we should definitely also try to find new model organisms that can answer new questions.
Yeah, and it also, you really need a whole organism to address certain questions. There's a lot of interactions between tissues. There's physiological differences, and you can't replicate that in a dish, no matter how hard you try. It's just not a normal situation. And there's many cases people make great advances by studying things in a dish, and it's really important. But when you go and look in the animal, you can get a different answer. So there's always going to be a need to study an intact animal. And as Anna says, you can do it very rapidly. Also, it's great. I think all the advances in molecular techniques are really amazing in the organoid technology, but still you can't grow a whole organ that's fully functioning in the lab.
I was just gonna, add that what is really amazing that anything can become a model organism now. In a way, for example, we are learning that naked mole rats, for example, don't seem to get cancer and live happy lives for very long. Bats have some special features. So because of the progress in genomics, being able to assemble genomes from DNA, we can very quickly learn new things about models that might be very suitable, animals that can regenerate well, or have a certain ability that might help us, improve our lives.
So how often do you discover new things in the research? Certainly I know from the publications side of things in communications, 80 to 100 papers published every year, but in terms of how the person in the street would interpret and understand those as new findings, some of them are small incremental steps, aren't they? But Julie, sorry.
Well, I think that what you say is right. So I think basically every day you learn something new. I mean, nearly every day. Sometimes everything goes wrong. So you have to be very kind of strong and not let the setbacks get to you. So things do happen and you have to overcome them. But you learn things very frequently. But a lot of them are incremental. And then suddenly, as you have these multiple incremental advances, they coalesce into something bigger and you come to a new understanding. So it's hard to say how often. It's kind of, you have these boom and busts of rewards, and it's really is gratifying. But to find some new understanding will happen many times during the year. And it's Eric.
Yeah, I was just going to add that science and research is not really a process, not like let's build a better car or let's try to find a better type of battery. I think a lot of this is sort of accidental discovery and unexpected progress, and as Julie already said, lots of setbacks as well. And in particular, in this sort of fundamental research that we are talking here about, it's often the case that you're working on something very specific and you don't really understand yet how this could be on wider relevance, and suddenly something opens up new and exciting that could reach all the way to potential new human therapies. It doesn't happen every day, but I think that's the kind of very special thing that we are trying to create and keep alive at the Institute.
Could you say a bit more about, what is that process going from a discovery that you think is really exciting, you've spotted something in the worm, and then taking that through to looking for therapeutics, because you are actually, you do have a spin-out company that's looking at this kind of issue, Eric?
Not that I have got tremendous experience, but I think I guess it is important that although we're doing fundamental research, really trying to understand sort of the fundamental building blocks, that we keep an eye out for where there are opportunities. And if one comes about, try to see if this can be developed further. And I think in the Cambridge environment, we're very lucky because there is a great tradition of spin-outs from the Institute, from the university at large, to try to help translate fundamental discoveries into new therapies or other applications. So I think watching out for it and then taking advantage of the environment where there's a lot of help there to, take you along on the path or tell you that, well, this is very exciting for you, but it's not really ready yet to take it any further.
Shall we say a little bit more about the other model organisms that we have as we featured in the tour video, and the sorts of things that are going on elsewhere in the building?
So we have, Eric and I both work on C. elegans. Eric works on many other organisms besides C. elegans, and I'll let him talk about those. But we have a big range here. As I mentioned, Drosophila. We have people working on Drosophila, studying brain development, for example, and also some cancer and how cells become different, what are called polarized. We have people studying, also someone studying mitochondria and how mitochondria work and how they divide, and that's also work in Drosophila. We have people, several groups working in mouse and studying how germ cells develop, how the lung develops, and also how those go wrong in disease. We study also Xenopus, this lavis frog. That also is very important in studying how genes turn on and off. That's a very nice model organism. We also have chickens. So really, and a lot of organoid technologies also.
So we do say that one of the benefits for somebody coming to work here is that they could quite easily, if they sort of generate a finding in one model organism, they might be able to then team up with people in another lab and develop that work by moving across into another organism and maybe sort of up through the evolutionary pathway there and test something out that might then get closer to, do we have a finding that could be relevant for human disease? And so it's really fabulous to have such diversity just within our institute here and just across 16 groups. Yeah. And so that's a real strength of the institute. That's
a really good point, Claire. And that does happen. I'd say a lot of the work that we do is collaborative. So labs get together and put their heads together, and really that the collaborative work is really, it's remarkable how much better things can be and what advances you can do as you work together with people with a slightly different system and a different point of view.
So maybe just one final question perhaps we could put to Anna, and just run back round to you and say, what was your career path here? How do you get to secure us this fantastic PhD position in such a wonderful place as the Gurdon Institute?
So I'm a third year PhD student at the moment. I'm on it's a kind of a rotation based PhD program that's funded by the BBSRC, which is the Bioscience and Biotechnology Research Council in Britain. But I did my undergrad at Oxford. I did a degree in biochemistry. But I think, I mean, there's quite a diverse, different number of people on my program, my PhD program, which is great. We've got plant scientists and people working in lots of different organisms, and it's great to work with them. But I mean, you can get to doing a PhD with any sort of undergrad degree in a science-related topic, right?
I think we're pretty much coming to the end of our event then. If there are no more questions from our audience, I'll bring the event to a close by saying thank you very much to all of our panel. Also, I'd like to say thank you to Peter Williamson, who's provided the IT support for us today. Thank you to the audience and for your questions and discussion. And we hope that in the future we can invite you to events at the Institute, but it has obviously been a real advantage of an online event that we can speak to people all around the world. So there's been an upside and a downside to the COVID situation. All that remains to say to everyone out there, keep safe, and goodbye from the Gurdon Institute today.
Goodbye. Thank you.